Good afternoon. Good afternoon, everyone. Happy New Year and welcome to MitoAction's monthly expert series. Today, Dr. Alejandro Dorenbaum, Chief Medical Officer with Reneo Pharmaceuticals, is joining us to share the recent results from the STRIDE study for primary mitochondrial myopathy, or PMM. Although the results were not what we all anticipated, MitoAction wants to ensure that we provide an opportunity for Reneo to share the details, lessons learned, and what this means for our community. Our journey involves not only celebrating successes, but also acknowledging setbacks, as they are integral to the advancement of therapy development. We are honored to have with us today Dr. Alex Dorenbaum, the Chief Medical Officer of Reneo Pharmaceuticals. Dr. Dorenbaum will provide a transparent and insightful discussion surrounding the recent clinical trial that did not yield the anticipated results, but despite these setbacks, our commitment to progress remains unwavering, and this presentation serves as a testament to our dedication and the community's dedication to our patients and families. Today's presentation will be recorded and available on MitoAction's website in the coming days, as well as on our podcast through Apple Podcasts, Google Play, and Spotify. We encourage you to ask questions throughout the presentation using the Q&A feature on the bottom menu bar of your screen. If you're calling in via the phone, feel free to submit your questions to us by email at info@mitoaction.org. And we will do our best to get through as many questions as possible throughout the presentation. Also, feel free to raise your hand so that we know that you have a question that you'd like to come on and we can have you turn your camera on and ask. So please join me in welcoming Dr. Alejandro Dorenbaum.
Thank you very much. I'm going to share a few slides, so let me see if I can get this going. Is that showing STRIDE study top-line results?
You're good, yes.
Okay, well, let me start by saying that these are top-line results. These are just the key endpoints that were evaluated in the STRIDE study, and we are still in the process of finishing the review of all the data in the trial. But these results are very significant because they are the ones that would be used as the primary evidence of safety and efficacy for the trial. And I realize it's a little early to talk about the entire study with all the results comprehensively because we're still doing that analysis. And but we felt it was very important to share with the community what we've learned so far. So this is a very brief summary of the finding of the trial. The STRIDE study did not meet the primary endpoint of the 12-minute walk test or the secondary endpoint of fatigue that was evaluated with the PROMIS fatigue instrument. We established that all the participants in the study were correctly dosed in the study according to what they were supposed to receive, the randomization, by using plasma drug levels. So we know that the patients that were supposed to get medicine had levels of the medicine in the blood measured, and the patients that received placebo did not have drug levels in their blood. So there was no error made in terms of who received drug and who didn't receive drug. This was essential to look at the quality of the trial to ensure that the study was done with good quality and we can trust the results of the study. Also, interestingly, we observed slightly more mild to moderate side effects, adverse events in the mavodelpar group with no obvious safety signal. This is important because what this is, and by the way, these adverse events that we saw are very similar to the ones that have been seen with other medicines that are similar to mavodelpar, PPAR delta agonists. And what this suggests is that mavodelpar has activity in the body. It works in the body and it does what it's supposed to do, but it didn't result in improvements in the endpoints of the trial. And then the last thing I want to say is we did a comprehensive subgroup analysis, including multiple parameters, genotype, phenotype, demographic characteristics, and I'll share a little bit more about this in the next slides. and the baseline disease severity and the geography of the patients, where they were enrolled, in which continent, and it does not disclose any signals of efficacy response. So we weren't able to establish that, for example, a specific group within the patients that entered into the study were more likely to respond or could be treated with the medicine as opposed to other patients. These are the demographic and baseline characteristics of the patients in the study. Overall, we enrolled 212 patients. Actually, there were 213 patients enrolled in the study, but one patient that was enrolled very quickly we realized didn't have the PMM and had to be removed from the study before that patient even received the first dose of the medicine. So the patient was in the study less than 24 hours and was removed from the study and did not receive any medicine. So in essence, there were 212 patients, 108 received mavodelpar, Reneo 1, and 104 received the placebo. As you can see from this table, the mean age of the patients was in the mid-40s, about 46 years old. And the group that was most represented in the study was the patients between 46 and 64, between 46 and 64 years old. As we have observed in other clinical studies of PMM, the majority of patients were women, and there weren't as many men. We've seen that in other PMM studies. It's not very clear to me why that happens, because the prevalence of the disease is similar according to the books in males and females, but women tend to participate more frequently in studies of PMM, and that was true in this study as well. And you can see this, if you recall, this is a study that was done in the post-COVID era. We had a lot of difficulty enrolling patients in the US, and instead we were very successful in enrolling patients in Europe. You can see that about 3/4 of the patients, 76.9% of the patients, were enrolled in European countries, and only 15% of the patients were enrolled in the US. And we've seen this happen after the pandemic, that the clinics in the US have had trouble putting together all the organization that is needed to enroll patients effectively in clinical trials. There might have been other reasons why patients may not have been enrolled in the US, competition with other clinical studies, or other reasons why clinicians or the researchers or the patients didn't elect to participate in North America. But that was a struggle for us. We had anticipated that we were going to enroll more patients in the US, and it just didn't happen after the pandemic. So just to give you an idea of what we did, we did 3 types of analysis for the data that we looked at: the full analysis set, the modified full analysis set, and the per protocol analysis set. And this is very important because it talks to the robustness of the results that we achieved in the trial. The full analysis set is intended to study all the patients that were enrolled in the study and have at least one measurement of efficacy. And you can see these are all the patients that entered into the study. And if a patient is missing a result, let's say at week 24 at the end of the study, then we do what is called an imputation. And you can see there the pattern, mixture, multiple imputations. So all these full analysis sets include all the patients that enrolled in the study. And if a patient doesn't have a result, then we give that patient a result that is the worst possible result accomplished in the study, based on a formula that is negotiated with the FDA. This is the analysis that the FDA likes most, because the FDA wants to consider that the patients that don't complete the study may have not completed because they had a bad outcome, or they couldn't continue in the study because their disease continue to advance, and therefore, generally, when patients are removed from the study, it is considered that the reason why they left the study might have been because the medicine is not working. Then we did a full analysis, a modified full analysis set, in which we look at the patients that completed the study, but we didn't do as much of the imputation. And we said, okay, let's just look at, for the patients that have data, what really happened in those patients without penalizing the analysis, without affecting the analysis with those patients that didn't complete the study. And then the per protocol analysis is just those patients who did the study perfectly well, meaning these are patients who entered the study perfectly well, who reported that they took all the medicine, that they didn't have any issues completing the trial, did all the procedures in the study according to the protocol. Everything went according to the book. And that's a population in whom you would expect to see the best possible result, because everyone adhered perfectly well to the study and did everything exactly like it was supposed to be done in the study. And you can see here that, for example, in the full analysis set, we look at the data in 7 different ways. The modified full analysis set, we look at the data in 2 different ways. And in the per protocol analysis, we look at it in 2 different ways. So in total, we did about 12 different analyses of the data to try to figure out, is there anything wrong in what we're observing? Is there anything that we can learn, or are we comfortable that the results of the study are reliable? And I have to tell you, we did not, all of these analyses did not show any difference, major substantive difference in terms of the outcome of the trial. As I said earlier, we explored the response in the 12-minute walk test and in the PROMIS fatigue score in multiple subgroups. We wanted to know, do patients who are older or younger respond better? Because patients who are younger probably would have less advanced disease. We looked at differences between males and females. We wanted to look at differences in race, but the great majority of patients enrolled in the study were white, so we only were able to look at the white population, and there were not enough of the other patients to be able to do an analysis. We also looked at differences between the patients that were enrolled in Europe and in North America, and we looked by mutations, patients who had the most common mutation, the 3243A to G mutation, the MERV mutation, the mitochondrial deletion syndromes, and then other mutations piled together. And we look at not only the mutations, but also the syndromes, patients who were enrolled who had a diagnosis of CPO or MELAS or MERV or other. We also look at the severity of the patients at entry into the study. We wanted to see, do patients that walk less than 500 meters, for example, or patients that walk more than 500 meters, do they do better in the study or do worse in the study? And the same for the PROMIS score test. We use 60 because PROMIS 50 is the center of the data. 60 means that the patients are 1 standard deviation worse than the mean of the population, and that's why we use the 60 number there. And there was a concern that vitamin D status, that patients who have low vitamin D may not perform well in the study, and that's why we also looked at the data based on the vitamin levels above or below the lower limit of normal in the trial. All of these analyses did not show any specific group that might have had a better response. There were so many other tests that were done in the study, but those are not the top-line results, so we don't have those yet, and that's why this discussion is very limited. And I anticipate that in the next weeks and months to come ahead, we will be able to share all of the results. We're committed to do that. But I want to conclude this presentation by saying this. we find no obvious reason to doubt the quality in the design and in the execution of the STRIDE study. The STRIDE study did not meet the primary or the secondary endpoints. And as you know, this was the pivotal trial for our program. We did a comprehensive subgroup analysis, did not show any signs of efficacy. And given the results of the STRIDE study, we see no viable path forward for mavodelpar in PMM. So we are suspending the PMM program at Reneo. And although the results of the STRIDE study were negative, the data generated are vitally important to the scientific community, and we will make the data available once the final analyses are complete. We think that a lot can be learned from this program that can help design better studies in the future, and that can give other drugs and other interventions the opportunity to succeed in PMM. And last, I want to say that we're really immensely thankful to the patients, the families, the healthcare professionals, this organization who participated in the mavodelpar PMM development program and helped us execute the program. On a more personal note, I can tell you that many of us at Reneo are really sad about the outcome of this trial. We really thought that the idea was a very good idea. We thought that mavodelpar would be a drug that would improve the mitochondrial function in patients with PMM, and I'm personally very saddened by this result, and we will do whatever we can to continue to support the community and find new ways to treat this disease. I'm going to stop sharing here and try to answer questions if there are any.
Thank you so much, Dr. Dorenbaum, for sharing from your heart, sharing the research that you guys have done. There have been several different questions that have come in that I want to ask, but I want to start with, you mentioned that even though the data isn't what you expected, that it's still vital to the scientific community. And so I'm curious if you could expound on that just a little bit with the things that you learned that you know that will continue to progress as we search to try to find medicines for a mitochondrial community.
Yes, of course. We've been having these discussions and chats with some of the doctors that follow patients with PMM. And as you know, when you develop a medicine for a rare or difficult to treat disease where there are not many patients, often we don't know very well what to expect from patients that are entering into a study. So this is what we call in medicine the natural history of the disease. What really happens to patients over time if you watch them, if you observe them over time, and how does the disease progress? One of the difficult aspects of designing this study is trying to figure out what would happen with the placebo arm. What would happen to the patients that don't receive medicine? Are they going to improve in their 12-minute walk test because they have a placebo effect? Are they going to get worse? If they get worse, how rapidly do they get worse? How rapidly do they lose faculties and ability to do activities? All of that information is very well known for big diseases where there are many, many patients that have been followed for many years. In cardiology, for example, you may have heard about the Framingham studies where a whole town, the town of Framingham, was followed for decades. And people have data on what really happens to patients who have slightly elevated cholesterol or who have slightly elevated blood pressure, and what's the difference between males and females, and what's the difference between white and African American people. So all that data is available because these patients have been followed for a very, very long time. So I think we have an opportunity to look at the patients that participated in this study, especially the ones that received the placebo, didn't receive medicine, where we know there is no intervention, and follow those and really assess that data very carefully. And that will give us more substantial, more strong data that others in the future can use to design better clinical trials. Another thing we did in this trial, and I want to address it because I still think it is valuable. I don't know if you recall, but in this study, we used a 12-minute walk test instead of a 6-minute walk test. And we do believe that the 12-minute walk test may be a better tool to use in patients with PMM. And that's something that when we analyze all that data, we may be able to at the very least, conclude what can be expected in a 12-minute walk test for patients who are participating in trials with this disease. This is the first time where 200 patients were treated and were included in a study, and the 12-minute walk test was used. So I think that now we have what we call normative baseline data that can be used in future trials that I believe they may be able to discriminate better the results of the patients. Another aspect of it is whether to use the 12-minute walk test or not, because we did in this study several other endpoints. In this study we did a 12-minute walk test. We did the PROMIS fatigue instrument, but we also used the MFIS, the Modified Fatigue Impact Scale. We did several subscales of the SF-36, which is a quality of life instrument that measures fatigue, vitality, and with general health. We also measured impact on work and productivity activities. We did the 30-second sit-to-stand test. And one of the things we haven't analyzed very thoroughly yet, because it's not in the top-line results, but was very interesting, is we asked patients to use a pedometer to measure general activity at home. And I think that data will be very interesting, will be very important to analyze and to compare to normal individuals and see what is the impact of the disease on patients with PMM, and how does that affect them. So I believe that will be very valuable data to look at, and what happens on a day-to-day basis with patients with PMM. How much do they really move or don't move? And analysis of pedometer data is not easy because there is a lot of noise in that data. But hopefully we will be able to look at it carefully and make some sense of it, and that may help as well in the design of future clinical trials.
That's really, really helpful. I mean, just even how you explained, the importance of the placebo, because I think as a patient community, that the placebo test is important, and that sometimes we fall into the placebo, but just to hear that you can use that as natural history in order to be able to help with future scientific endeavors and collecting data in that way. And the walk test too. I was unaware that it was the first space that extensive walk test was used. So I think that's really helpful information. I appreciate you sharing all of that.
Let me add one more thing. Let me add one more thing. I've been in PMM meetings. We've been in meetings where I remember the community was very concerned about the use of placebo in clinical trials. And I recall one meeting where that was a major issue, that we were talking about why do patients need to go 6 months or 12 months on a placebo arm, and we're not this is not something that anyone wants to do. But if you look at the results of this trial, it's very interesting. When when we look at the results without knowing who was on medicine and who was on placebo, you could look at the results and convince yourself that there are patients who seem to be responding very well to the medicine. But at the end of the trial, what we realize is half of those patients were in the treatment arm and half of those patients were in the placebo arm. So basically, there was no difference between the two interventions, between getting a placebo or getting a drug. If anything, I think this trial should help the community understand that it is really important to use placebo-controlled trials, because otherwise we cannot tease out the results of whether a drug is really helping or not. People believe a medicine can be helping them when it is really not helping. So I think that, if anything, this study really strengthens my resolve to recommend that we continue to do placebo-controlled trials. Now, the question in my mind is more about figuring out what are the real good endpoints to look at. And to me, the 12-minute walk test or the 6-minute walk test is a test that nobody wants to use, but we all have used it because the regulatory agencies like the FDA and the European agency, the EMA, like this kind of endpoint. But it seems to me that one of the things I learned from interacting with the PMM community is that there are several issues that are of most concern to people. And I think that the issues that are of most concern as the limitation in ability to do activities of daily living and the fatigue that patients suffer. And I think that those are key endpoints, key issues that we need to explore, and we need to work on ways to better measure those changes. I think that will be very important.
Well, and I'm glad you brought that up, because a person put in the chat, they asked, what other endpoints do you think could be used in the future in regards to mito drug trials? And I saw some hearts go out there too, as you were mentioning the fact that, like, is this the only endpoint that we can really use? Do you have any thoughts on that question?
You know, I have to say, people much smarter than me already got together and sat down and talked about this. And there was a very nice paper written a few years ago about what potential endpoints could be pursued in patients with PMM. That's a publication that was written by almost all the experts that see patients with PMM. If there is any advice that I can give to this group personally, and I don't propose to say, I really mean it when I say these were much smarter people than me. I'm not trying to be humble. I have met them and I know they are smarter than me. So it's not that I can be smarter about this than your own doctors that see you. They are really good. They know what they're doing and they do it very well. It's just, I think what I would think is that there has to be a medium point where you work with an endpoint that can convince the FDA that the drug works, an endpoint that is important to the patient. You know, you're fixing something that I care about. Otherwise, I don't care about it, you know. And also an endpoint that is practical, that can be assessed easily. And I think that part of the problem is that many of the endpoints that we are using are not very practical. They're not very easy to assess. And I think we need to try to simplify matters as much as possible and really get to the core of the problem that patients with PMM have. And you know, there was, you know, the FDA cares about this, because if the FDA a few years back did a program called The Voice of the Patient, where they brought your patients with PMM and their families and their doctors and the industry to talk about it, and they published this document, The Voice of the Patient, that talks about what are the things that patients with PMM care about most. And I think we need to go to the basics, you know. I think we need to go back to the basics of listening to the patients again and trying to find ways to resolve their symptoms and measure that change in those symptoms and not focus on the other things. Thank
you. That's really helpful. Another patient asked, do you have any comments on the severity of disease? She is guessing that maybe higher functioning people were recruited for this study. And do you think that the medicine would work better for those who've progressed maybe more, instead of maybe it working better for those who haven't progressed quite yet? I know you mentioned you did some types of cross analysis with some of that. she suggested, like, what about those who couldn't maybe meet the 6-minute walk test? So some of this kind of goes back into like having different endpoints, like the endpoint is you couldn't brush your teeth by yourself before, but now you can. Like that might not be a 6-minute walk test, but still be huge in a patient's life. What are your thoughts about any of that?
So I think that some companies are beginning to do. So the question is, could this drug work in a more severe population or a population that wasn't studied in the STRIDE study? And the answer is very difficult. I don't know. I don't know because we didn't study it. So I don't know. I thought we picked a population that was sick enough, but that would still have enough reserve to be able to fix some of the problems. To me, that was very important. When we designed this study, we wanted to say, okay, what would be the perfect population to study? And in a 12-minute walk test, patients, normal volunteers usually walk about 1,200 meters or 1,400 meters. And in the baseline of our patients, they walked about 650 meters, which is a little bit less than half the distance a normal individual can walk. So these were patients who were quite affected in their disease. They cannot walk where a normal individual can walk, but they can still walk some. And that was very important because if a patient cannot walk, then you cannot assess if you're improving walk at all. So we thought we studied the right population, but we can't tell what would have happened if we had enrolled a different population into the trial. Part of the problem in doing a study, and this is again, this gets back to the statistics, is that in order to be able to do a study and have a good result of the study, you need to make sure that the patients are all very similar entering into the study. Because if they are very different, if there is what we call heterogeneity in the patients, then some patients may respond, others will not, and you may not see you may not see an overall response in your trial because the ones that are not improving are dragging the patients that are improving down in the overall results. So it's very difficult to do that. And that is best explored in early phase development. But the problem is in PMM, we don't have that many patients to be able to do many studies before we do a larger study. And so those type of questions are easy to answer when you have a disease like diabetes, and then you can say, okay, let's take the worst diabetics, the medium diabetes, the lower diabetics, and because there are 20 million diabetics in the US, you can do the studies very easily, but here it's very difficult.
So another patient is asking, so when the full data comes in, is it possible when the full data is analyzed that there could be any difference in the decision in regarding to suspend the study, or is it definitively canceled?
No, the study is definitively canceled. And the reason for that is because we looked at the key endpoints of the trial. And without those endpoints, there is no hope that the FDA would ever approve this program. So because there is no hope that the FDA would approve this program, it's not, how would I put it? It's not even ethical to continue to do the studies because we know this is not a drug that would be approved by the FDA based on what we already know. So it's as if you had a car where the engine went bust and you say, well, if we put more gas to the car, can we run it? But no, the engine went bust. So unfortunately, the results of the study are the key results that the FDA would look at to approve the drug. And because those results were negative, we know the drug will not be approved. So there is no point in trying to do it, in reopening the study.
So how does the closure of this study affect the FAOD study, which was using the same the same drug. Will Reneo continue to pursue this drug in the FAOD community, or are they both being suspended?
We're suspending both because if our thinking was that the perfect population to study was the PMM population, and the results of the phase 1b studies were very similar in PMM and in FAOD, at least for a subgroup of the patients with FAOD, not for all of the patients for FAOD. For example, when we did the study in FAOD, if you recall the fatty acid oxidation defects, there are at least 4 different diseases that have large fatty acid oxidation defects. One is called VLCAT, one is called LCHAT, the other one is CPT2, and the other one is trifunctional protein defect patients. And what we saw in that first study is that the patients with LCHAT responded very similarly to the patients with PMM, but the other patients did not respond as well. So we are concerned that the responses in that phase 1b were not even as good in general for the entire FAOD population as they were for FAOD, and yet, and they were very similar in the LCHAT patients. So we've decided not to pursue further in that population. We think that there may be other things to do with those patients that may work better than this treatment, and that's why we decided to suspend that program as well.
Okay, thank you very much. That's very helpful. Another patient's asking, she asked if you could measurements of VO2 max help measure severity of PMM patients?
This is a really complicated question, and let me address it. I've thought a lot about it, and it gets back to the basics of medical physiology. But I want to say yes. The answer is yes, but let me explain how complex it is. When you're doing VO2 max, what you're measuring is how much oxygen is your body using in order to generate energy. And the reason that is valuable to patients with PMM is because the way the mitochondria generate energy is by using oxygen. So when you're breaking down the fats in the mitochondria, the lipids in the mitochondria, you use oxygen, and that generates energy. So the more oxygen you use, more energy you're producing. And the less oxygen you use, less energy you're producing. Now, initially, when VO2 max was developed, when the test was developed, and this is how the test is done very briefly, you ask a patient to start walking as fast as they can in a treadmill, and you put a mask to measure how much oxygen is being exhaled. And if you are using the oxygen, very little oxygen will be exhaled. If you're not using the oxygen, more oxygen will be exhaled. So what you're trying to see is how much is being consumed by the body to generate energy. And you increase the treadmill as much as possible, and you get to a point where the heart rate gets to the maximum heart rate you can get. And I don't know if you remember, but each one of us, based on our age, we get a maximum heart rate. Mine is lower than yours because I'm older than you. As you get older, your heart rate doesn't go as high as it goes when you're younger. So how much oxygen you're consuming when your heart rate is at its maximum rate, that's what's called VO2 max. And that gives you an idea of how fit an individual is. The study was invented to evaluate cardiopulmonary function, because the main problem that exists in the world is the heart is not working very well. So if the heart is not working very well, it's not pumping blood to the tissues, and the oxygen is not arriving in the tissues, and it's not being used. But in PMM patients, the problem is different. Generally speaking, there are patients who have cardiomyopathy and their heart may not be working very well, but the majority of patients, their heart is actually working relatively well and even working a little harder than usual to make up for the problems that patients with PMM have. So the heart doesn't seem to be the major problem in the use of oxygen. In other words, the heart can deliver oxygen to the tissues. The problem that happens in PMM is that when the oxygen gets to the tissues, the mitochondria don't use it because they're not working well. So the oxygen returns and it's exhaled. So the VO2 max in patients with PMM is very low. And that is because their tissues are not using the oxygen, which is different than the heart not being able to deliver oxygen to the tissues. So almost everything we know about VO2 max is related to problems in the heart and in the lungs. And we don't know as much about VO2 max when the tissues cannot use the oxygen. I don't know if I confused the group, but the problem is, sometimes the problem is, can you supply oxygen? And the other one is, do you use the oxygen? And we know a lot about when you cannot supply the oxygen, but we don't know enough about when you cannot use the oxygen. But I think it's a good measurement in PMM patients because what it's telling you is the mitochondria are not consuming the oxygen to generate energy. So it could be a very good test. The problem is doing the VO2 max test is difficult, but it actually could be a reasonable endpoint that could be negotiated with regulatory agencies because the regulatory agencies understand the VO2 max test very well. And that could be an endpoint that could be considered in future trials, definitely. It's very difficult to do it. So it would need to be done with a lot of caution. And if a company decides to use VO2 max, I would recommend that they do everything in their power to make sure that that study is standardized very carefully so that it can yield the right results.
Thank you so much for all of that detail and breaking it down, because that made it very digestible and understandable. So, and thank you for the participant that asked that question too.
Very good question.
Yeah, no, these are important thoughts that come out. Like, that's one of the things that I love about these roundtables is that patients and clinicians ask such good questions, and together, if we ask the questions, hopefully there'll be more answers. Another question that came in is, if you were to start fresh, what are the top 2 to 3 things you would do differently?
I really think the study we did was well executed. I really think that when I look at the quality of the study was done well. So the answer I'm going to give you is a very painful answer to me. It may sound funny, but I wish I had had another drug to try. I really wish we get other drugs to try. I think that what this community needs to work on is pushing the companies and pushing the investigators and pushing the researchers in the universities to come up with other ideas. There will be a solution to this problem. There will be, I think that we've talked a little bit about this before. My background is not in neurology or neuromuscular diseases. I became a doctor treating primarily patients with immunological diseases. And when I did my training, the main disease that we were dealing with was at the very beginning of the HIV pandemic. And if you recall, back in the late 80s, early 90s, HIV was a disease that basically meant if you had this disease, you were not going to survive. And I'm a pediatrician. The children with HIV used to have a very poor outcome very early in life. Children did not last many years with that disease. And I can tell you, it's very, very, very sad to say this, but of all the patients that I treated when I was in the university treating pediatric HIV, I am aware that only one patient has survived. only one of all those children that I treated, only one of them is still alive. All my other patients, unfortunately, died due to the HIV. A child that is born today with HIV, if he or she receives treatment, they can probably live a normal life. That is impressive. And that happened in my lifetime. I was able to see that. And it took 20 years to do it, but it happened. I believe that the mitochondria are becoming very important in the research of medicine. I think that people are realizing that mitochondria are essential in many situations. I think that there is tremendous research being devoted to understanding the mitochondria and to understanding how we can improve the mitochondrial function. And I can see that with pressure from the community and with better understanding of how mitochondria functions, there will be a cure. There will be a treatment, if not a cure, that will be able to help patients. And I just think that I mean, the work that that you guys at MitoAction do is essential, because it will push the investigators and the companies to develop new treatments. I think that's the most important thing that we can do at this point. Push for new treatments to come into research. And then when the new treatments come to research, it's very important that the people that have the disease are willing to participate in the treatments, in the research, and so that we can quickly answer the questions. Look, none of us can promise that a result of a trial will be positive. But what we need to make sure is that we try as many things as possible so that we find that jewel that will be the one that will help patients. And if patients don't participate in clinical trials, we will never be able to answer that question. So I know it's stressful, it's difficult. You know, when patients participate in clinical trials, we do what we call informed consent. We tell patients, these are the risks, these are the potential benefits if you take part in the study. And when I read those risks, I say to myself, would I be willing to do this? Would I be willing to try this? But the bottom line is most of what we know in medicine is because people took a chance, and we have to continue to take a chance.
Well, I'm wiping my tears away. I just feel like that, I mean, all that you're speaking is truth, right? It's beautiful truth, it's hard truth, and it's totally truth for our community. So at first, I just want to thank you for being vulnerable and being honest about your experience as a clinician working in the HIV community, because it is those moments that bring us all hope. My sister had a rare form of childhood cancer, and in the 80s, and to see where it comes from, it brings me hope for my son who has this rare disease, right, in the 2000s. And so I think there's a lot of hope that we see, and we really appreciate your vulnerability in that and being honest, and also just being honest about the drug. Like, I feel like you're very clear, like, you know, it'd be helpful if we had more things to try, you know, because this isn't the answer. And I think sometimes that's a hard pill that has to be for the scientists to swallow, who've put in all this time and effort, for the patients too, because, you know, there's hope that you place in it. And yet, if it doesn't work, you know, it's more important that something that doesn't work gets passed, right, than something that works. So putting our efforts in the right place. I see your heart in that. So thank you so much for having such an honest and pure heart and your explanation with all of these things. There are lots of more questions that are coming through, and I know that we are getting close to the end of our time. So I just want to let others know that there are a couple questions that came in that are very similar. I'm going to try to combine them just for sake of time. Sure. So a lot of people are asking, you know, you mentioned when you talked about the FAOD study, but also about the primary mitochondria myopathy study, that maybe there'll be other interventions in the horizon, or maybe that we can use the things that we learned from this study for future things on the horizon? Are there trials that you know on the horizon that and you might not be able to share that information. I don't know, but patients are
but for
asking,
example, I mean, again, I've been thinking a lot about the implications of our trial, and if you recall, there is another company that has another PPAR delta in development in PMM. Now, one of the questions that might be asked is, could this drug work in patients with, what is the difference of the response, or would this drug work in patients who have mitochondrial versus nuclear gene defects? Because we only studied mitochondrial gene defects. We thought that was the right population to study, but we never studied the nuclear patients. So one of the questions that could be asked is, could this drug be beneficial in patients with nuclear gene defects? We haven't answered that question. And if I was in the other company developing a drug in the same space, I would say, well, the question we would be willing to show our results to the other company we know is Astellas. So I think Astellas needs to look at our results, and then they need to make a decision. What do we do next? And an option would be to say, okay, we're going to focus on the nuclear patients because those have not been studied, for example, or that could be a potential answer. But again, I can't tell Astellas what to do and what not to do, and they may have good reasons to do it or not to do it, and I think that it would be worth discussing it with them. But I think that would be an important implication for what they are doing. Generally speaking, when I think about disease, and this is just a general concept that I think of, I think that disease is a very difficult state. I mean, in general terms, and this is again after many years of dealing with very sick patients in my life, I come to the conclusion that disease is sort of, I hate to use this analogy, but let me use it anyways. I think that disease is like a freight train coming at you. You know, it's not, when the body has many mechanisms to overcome problems. The body fixes many problems very well. When the body cannot fix a problem, it's because it's really bad. It's like a freight train coming at you. And I think that the interventions that will need to be done for this disease are not just stop signs that you try to put to a freight train, but big interventions that will need to be very powerful to control and to improve the mitochondrial function. So I think we need to be more in a sense, the treatments that should be developed for the disease, in my opinion, need to be very aggressive treatments that will really try to impact the mitochondria very strongly. And again, the experts in mitochondrial function will be better at coming up with ideas than me for this.
So, okay, so this is more a specific question, and I hate to kind of like meander
Sure.
back and forth between.
Sure. No, don't worry.
But I do, so this patient asked, was there a common endpoint that showed improvement in the test group that differed from the placebo group? In other words, were there any patterns in the test group that were not present in the placebo group?
No, and that is a big problem. That is the big problem, that we don't see a clear endpoint that would be more likely to be beneficial, at least in this trial. So that is a big problem. Yes, when you look at the results for all of the endpoints that I've looked so far, they look very similar whether you receive drug or whether you receive placebo.
So you have some comments coming in, people thanking you, people saying they're willing to participate in future trials. So please, continue on. So I want to make sure that you hear that from our patient community. And also there was a question that came in about young children and studies in regards to young children. And I'm just curious, I know that this study, I believe you had to be older than 18 to participate. Can you talk a little bit about the complications of studies with younger children and how that's typically approached?
Definitely. So the PPARs the PPARs, PPAR delta agonists, and all the PPAR agonists are medicines that stimulate genes, activate the activity of those genes, and those genes affect the mitochondrial function. But they can affect other genes as well. And the concern that exists in children is that as children are growing, if you affect those genes, you could affect the growth and the development of the children. So in order to move forward in children with these medicines, we need to acquire a lot of experience with these medicines before we can give it to children, because we could affect unintentionally their growth and their development, because these interventions are difficult. But there are other therapies that have been, there is a ray of hope, for example, in PMM. There is the TK, one of the mitochondrial depletion syndromes, as you know, is being treated, with a combination of medicines that seems to be working a little bit better for these children. And I think that children pose a very unique problem. For example, we were studying the 12-minute walk test, and the question is, what would happen to the 12-minute walk test in a teenager that is developing muscle mass? You know, it's very difficult to evaluate a moving target. An 11-year-old cannot be as physically active as a 14-year-old or a 16-year-old. So putting the adolescents in a trial makes it very complicated, because they're not only changing in their disease, they're changing in their body. They're becoming taller, stronger, more muscle mass, and that makes it very difficult to study them together with the adults. So I think the studies for children need to take into account many other things. That first, that the children are developing. Second, that they may be changing significantly. And third, a very important issue is that when you make a decision to participate in a trial, you make that decision, but a child may not have the ability to make that decision very easily. So everything we do when we treat children has to already have a balance that tilts towards benefit. You need to have some evidence that the drug may help the child in order to put the child at risk of participating in the study. So it's very complicated, but the ethical aspects of studying children are very, very serious. And but on the other hand, it's not ethical to ignore children, and children need treatment as well. So it's very important to study the children as early as possible.
Well, thank you so much, Dr. Dorenbaum, for your time. Thank you so much for your vulnerability and all of your answers. This is a super, super hard topic, and it's a super, it's a challenging thing, I think, to break down, but you've done an absolutely amazing job, and we just so appreciate you and all of your work and all of the efforts that Reneo has made, and all the strides forward that they've made with the data that will continue to come through. Kyra, I don't know if you have anything else you'd like to add.
Yeah, I just wanted to echo everything that Stephanie says. I know this is a tough conversation, but it's a necessary conversation. And in the spirit of transparency, education, and resiliency, right, we want to make sure that we continue to have these conversations so that the community understands exactly what's going on and how we can all continue to take an active role, right, and keep hope on the horizons. Because even though we have this setback, we still are continuing to move forward. And there are so many people that are working hard to support this community. And we have to stay positive, and we have to keep pushing forward. And so we want to make sure that we're all working together to do that. And as unfortunate as the results are, we know these efforts will help to pave the way for future advancements as we continue to strive towards effective treatments and improve quality of life for those affected by mitochondrial disease. Dr. Dorenbaum, we're so grateful for you and your team, the entire team at Reneo Pharmaceuticals, and for your commitment and the work that you all have put in. And we know this is not the outcome that we all wanted, but we do hope that you know how grateful our community is for the progress that you did make and your commitment to this community. And we will forever be grateful for you, for that. So we appreciate that.
Thank you very much. Thank you.
Yes, thank you, everyone. And again, our presentation will be recorded and we'll have it up on our website and on our podcast channels in the next few days. And also, Reneo will also be on the town hall meeting next Friday to also give some more details. And we hope that you'll join us because again, we need to stay focused and positive on what's ahead. And we're going to focus on that next Friday and hope everyone walks away feeling encouraged and hopeful with all the work that's being done for this incredible community. So thank you everyone for joining us today. We appreciate you.
Bye.
Take care. Bye-bye.